are going to have some application of EzScope that we would like to share to you. And for today's webinar, we would like to divide into two parts. For the first part, will be our product specialist, Frank. He will introduce the purpose of using live cell image system and some case studies for use. The second part, we are very glad to invite the real user, Professor Wang, who is specialized in virus research and will introduce how his virus infection test research was improved by the digital cell imager. So at the beginning, please check your device that you can hear our voice very well. And during the meetings, if you have any questions, please check the downside of your monitors and you can type words in the Q&A rooms. Okay, so now we are ready. So let's start the course from Frank. Hi, Frank. Please say everybody hello.
Hello, everyone. Hello, everyone. Thank you for joining us today. My name is Frank Wang, and I'm a product specialist for the EzScope. And at the beginning, I would like to start with introducing you the concept of the live cell imaging system. So I think all of you here are cell-related researchers, and some of you may familiar with the live cell imaging system, but some of you may not. So I want to start with the concept of the live cell imaging system in this part, and hope to provide you with a few key points for choosing the system. Okay, so I like to start with this comic because it's very interesting, and I think if you are cell-related researcher, I think many of you may encounter this problem more or less. So in this comic, the professor asked to record cell images every single hour for total 48 hours. So it's very scary, but I have to say that I think the professor is not personal, okay? So because sometimes you really do need this kind of data. And so if unfortunately you need such a data, then you will need to consider some problems. So next parts is some few things you have to consider if you encounter this situation. So the first one is if you want to capture cell images every hour for total 48 hours, you have to move the cell samples in and out of the incubator. And if you do this, you are causing the culture environment keep changing. Like you will lose the CO2 and the temperature dropping. And these two phenomena will cause you the cell growth will be affected. And because the cell growth time is calculated in days, so if the observation time interval is too short, it will cause the labor burden, right? Because you have to stay with the microscope, you have to stay with your cell samples. So the labor burden is very huge. But in the other side, if the observation time interval is too long, the critical moment may be missed. So you have to balance it. But two of these methods will prone to human error because you don't have the timer like machine, right? And finally, you will want to observe multiple groups in one experiment. So it's better, it's best to control multiple systems at the same time. Okay, so this is some points you have to consider if you want to do such a cell imaging recording. Okay, so in order to solve this problem and achieve your experiment, you will need to you will need a live cell imaging system. So what is the live cell imaging system? Based on the Wikipedia, you can find the live cell imaging system is the study of living cells using time-lapse microscopy. And take these pictures, take pictures of cells in every certain time interval. This is the time-lapse microscopy. Okay, so if you want to take this kind of, we call that time-lapse pictures, you will need a microscope, and you will need an incubator for keep cell alive, and you will need a timer, and you need a camera. So combine this, it's a live cell imaging system. Okay. So now in the market, there are two major systems, two concepts of this system. So the first one is incubator on microscope, and the other one is microscope in incubator. So let's check out the first one, incubator on microscope. This is a microscope with an add-on incubator. So you can see many accessories needs to be added, including temperature control accessory, gas control system, and the other accessory have to add on. And the advantage of this system is you have a high image quality because it's a regular microscope. But the bad way is the price is very high and the structure is very complicated, and the cultural environment is very easy to change. So this is our first concept, the incubator on microscope. And for the second one is microscope in the incubator. So this is a specialized cell imaging instrument, specially designed to be placed in the incubator. Because incubator is it has a high temperature and high humidity environment, so this kind of specialized design can be placed inside incubator with a very stable environment. And the price is relatively lower than the microscope on, incubator on microscope. And you can save the space because you can see you can put the system inside incubator and there's a lot of space left. Okay, finally, you will want to consider several key points when you're choosing a system. And I'll say that the first is, of course, easy to operate. And it can be operated remotely and without having to open the incubator all the time. And the expandability. And you want to be able to replace different magnification lenses and the mechanical stage, and whether it can control multiple systems at the same time. And finally, the price, of course. So Blu-ray Biotech, we launched the live cell imaging system last year, the EzScope 101, and its feature of time-lapse photography, continuous observation, and the digital recording, plus the ability to be placed inside incubator, make it a great help to the user in the field of cell research. And it can also connect one PC to 4 units at the same time, which means you can control a full imaging system at the same time. So we hope this product can help researchers in continuous recording and observation. So this is my part, and thank you.
Okay, thanks for the introduction from Frank. So now we can check whether does anybody have some questions. Let me check the Q&A rooms. Okay, so it seems, okay, so if you have any question, just type the words in the Q&A rooms. So we will check the questions. Okay, let's enter into the second part. So the second part will be the real user, Professor Wang. Hi, Professor Wang. Could you please say hello to everyone?
Hi, everyone. Nice to meet you.
Thank you, Professor Wang. Okay, so Professor Wang is ready to share his research for everyone. So now I would like to turn for Professor Wang. Hi, Professor Wang. You can start to share your research of live cell imaging.
Hello, everyone. I'm Xiangyu Wang. I'm the assistant professor in National Pingtung University of Science and Technology in Taiwan. Today, I want to sharing about the live cell imaging that we use it to perform the continuous imaging recording for the different cell clones in various infection tests. In our laboratory, we are focusing on the study of virus and cell interaction. We observe the cell response in the virus infection condition, and we use this kind of knowledge in the vaccine production area. And the purpose here is try to looking for the highly sensitive cell clones in the virus production. In the industry, the highly sensitive cell is one of the keys in the process design. So how to isolate highly sensitive cell clones is very important. However, this is time consuming and a lot of work needed to do. You can imagine that you can infect the virus that for into different cell lines or into different cell clones, and then you try to recording the imaging results, or you can isolate the supernatant for the virus titer. And if this cell line is 100 or 200 clones, so it is a lot of works. In generally, we culture cell in the flask or in the dish, and then after infection, we observe the cell CPE under the microscope by using of the recording the imaging by camera. Of course, by this process, you can only sampling or you can only recording the imaging on some specific time point. For example, 20 hours or 24 hours. And this kind of results, it will lose some continuous or some specific cell response during this time period. So now we want to show you the data that we recording for the different 3 cell clones. They are derived from the BHK21 cells. And the upper panel is the cell-only control group, and the lower panel is the virus infection group. And we infected the virus into the cell at the very beginning. Now we start trying to see the video. After virus infection, the cell still can grow more and more. And in the after 24 hours, you can see a lot of CPE shows up in the different cell clones. And however, some cells grows more and some cells did not. Of time, you can see the very different result between these three cell clones. You can see here, a lot of cells survive, but a lot of cells have CPE. Here, it shows CPE, but a lot of cells still alive. Here, you can see that a lot of CPE, but less cells survive. And now it's get into the 48 hour. You can see the very difference between the three cell clones. Here, a lot of cells survive. And CPE, yes, it has CPE. Here, just between these two groups, here, this cell is very sensitive to the virus infection. And then to the final, you can see that almost all cells was died in this group. Here, some cell survival. Here, maybe a lot, but yes, it shows CPE, but many cells still alive in this group. So, by using of this data, we can very easily observe the cell response to the virus infection and the results within the virus infection process. By using of this kind of data or knowledge, we can manipulate the process or the production process in our bioreactor or the manufacturing design. So this is my short presentation. Thank you, everyone.
Okay. So thanks to the Professor Wang's research and sharings, it is really interesting and let us know to understand that the virus-infected cells are morphologically different. And now we are entering to the final part. Our product specialist, Frank Wang, would like to share some applications from other users. So let's welcome Frank again.
Hello, everyone. Thank you. Okay. In this part, I will share a few more application cases which are provided by our customers. Okay. So before we go into the application cases, I would like to spend some time to see the EzScope 101 and its some features, and then it's the 4 application cases. So this is the EzScope 101 with the XY stage. So we have XY stage, and we have 10x and 20x exchangeable lenses. Okay. Okay, then it's I want to next part I want to share with you the latest EzScope application and how it can assist our research. So the first case is from a university cell culture experiment course. And there are students in this class. They were learning how to culture cells and use to further analysis like cytotoxicity experiments. And they use the EzScope 101 to observe the process of the cell growth. Okay, so I think this application is very simple, but for the beginners, there is very important to understand the growth characteristics and the morphology of the cell you cultivate. So what we will looking at is a 549 lung cancer cell and HOS bone cancer cells. So in the left, you can see there's a A549 cell line with 10x under 10x lenses with the interval 2 minutes for a total of 24 hours. So you can see cell expanding and proliferation. And in the right is the HOS cell line in the same condition. So you can see there are two different cell type, and they have shown a different characteristic that A549 cells are much more mobile than HOS cells. Okay. So this application case is very simple, but I think if you in your laboratory course, you will need this kind of instrument in your class. If you need a long-term observation of cell or tissue samples, you will need a live cell imaging system. And the next case is also come from Professor Wang's lab, and this is the cells and the virus infection. And they observed the growth of the cells in the left. They waited until it reached a certain level before infecting the cell with the virus. So this is the cell growth observation. And in the right video is the cells, and they infected with the virus. So you can see a lot of small black dots forming. So you can see that this viral infection is progressive and cause cell death. So it's important to them to identify this characteristic of the virus when they can determine it in the dynamic video. Okay. So this application explains that if you are working on pathology experiment, especially for virus, you want to determine the progress, the process of the virus infection status. You can use this kind of cell imaging system and to determine the morphology changes. And the case 3 is very interesting that this is come from our friend Dr. Chi. Dr. Chi's research is going to be published, and it's about the result of the migration assay experiments. But the principle is as follows. This is from Dr. Chi's research. He does the transwell assay, and this is a kind of commercial kit. And there is transwell inserts, and you put it in media, and there the cells grow on inside the transwell inserts. And they want to observe whether the cell will pass through the permeable membrane to a lower compartment. And finally, they will measure the migration ability of the cells. However, the reviewer, they think that they ask for the flat observation of the cells images because the transwell is an endpoint. It's an endpoint moment. It's fragmented, and it cannot be confirmed that most of the cells move to the lower compartment or proliferate after moving to the lower compartment. So that's their question. So the quantification of migratory cells using this method is based on the endpoint measurement. So you don't know what happened between. So Dr. Chi found a migration slide kits from Merck Minipol. The migration slides is especially designed for video microscopy assay. So at specific time interval, the observation area can be acquired at this area and allowing real-time monitoring and quantitative measurement of the cell migration. So they need to observe this area, but Dr. Chi needs a tool to help him observe and record this kind of field of view. So Dr. Chi found us, and he used the EzScope 101 to observe these slides. So in the video in the left shows the cancer cells' ability to migrate. So you can see the cells is moving from the top to the bottom here. So it shows the migratory ability. And the video on the right is the cells, and the migration gene was being knocked out. And you can see the cells, they're still moving, but they are moving without directions. Okay. So this is a very important tool for Dr. Chi, because he just a few days ago, we just received the news that this paper was being published. Okay, so for the migration assay, you need a real-time monitoring, you need a dynamic determination. For the migration assay, this is a very good, the live cell imaging system is a very good tools for you. And the final case is a 3D spheroid. This is a cell-made container used to cultivate 3D tumor sphere. And you can see that the cell have successfully formed tumor sphere after 12 hours of seeding. But what happened in the between is unknown. Okay, so if you use live cell imaging system with time-lapse capturing function, you can go back to this moment, and even you can even convert it into an intuitive video. You can see there's a small cluster and then merge to one big sphere. So this is a very good example. When you need a live cell imaging system, you can capture images every maybe 30 minutes, 1 hour, and you can convert it into a video. Okay. So the above is the application cases using the EzScope 101. So in summary, the EzScope's feature of time-lapse photographing and the continuous recording is very good help to the cell-related research. I think this, I hope this sharing today will help you better understand of this kind of system, and hope this kind of product will help you on the research. Thank you.
Okay, Thanks for the introduction from Frank and share some application from the end user. Now we got some questions from our Q&A rooms. So let me pick up some question and answer to you. Okay. So the first one is, what size cells can the EzScope image? So, Frank?
Yeah, I think that we use the 10x and 20x lenses. So if you can if you use your microscope with under 10x and 20x, you can use EzScope. It's okay. Okay. So normally cell size is maybe around from 20 to 70 micrometer. That's okay for the EzScope. And does it have a moving stage? Yes, we have XY stage, but it's for manual operated. Okay, how many instruments can be connected to one computer? The answer is 4. We can connect 4 units to one computer.
Okay, so we have many requests from the EzScope. Now I am pick out the final ones. So for the residual questions, we will sort out all of them and reply to you by mail individually. So let me check the final questions. What software is used? And did you need Wi-Fi to transfer the data? So Frank, could you please help us to answer these questions?
We are connected, the EzScope 101 is connected to the PC, the computer, so all the data will saved in the computer. Okay.
Case. Okay, so now we got many questions right now. Wow, so many about the price, about the delivery time, about the lead times. So let me pick out the final ones. Can the image be observed via internet from home or other location than the lab? So Frank, please.
We connected the live cell imaging system, which is the EzScope 101, to the computer. So this computer is Windows. If you can find any remote monitor application and can install on this personal computer, you can view this. You can view the image of the cells from your laptop in your home. That's okay. That can achieve from the remote monitor application.
Okay, sounds very good. It saves time and saves effort. So thank you everyone for attending the webinar today. So in the future, once we get more application about the EzScope, we would like to share to you by mail. And I want to say thank you again and say bye-bye to you. Bye-bye.